So in this second part of the second lecture, we'll be talking about multifactorial and complex trait. So some diseases and phenotypes in general are not caused by a single gene. Rather, they are caused by multiple genes. And these diseases are known as polygenic diseases or phenotypes. Now, the effect of these genes is accumulative, meaning that there is no dominant or recessive allele, rather that each allele contributes something to the phenotype or to the disease. Now, if there are few genes causing a certain phenotype, then we call this oligogenic traits. Okay, but if there are many, then we call them polygenic phenotype or polygenic trait. Now, when an environmental factor is involved in the appearance of this phenotype or in affecting genes in general, genes and gene expression and gene activity, then we call this phenotype or disease or trait multifactorial. Okay, sometimes it's the environment, like for example, exposure to sun, deficiency of certain vitamin, or sometimes it can be a pathogen, like viruses or bacteria inducing autoimmune diseases, which are also multifactorial. Now, so we can classify such disorders into two types. We have the congenital disorders, these that appear at birth, and they involve they involve multiple genes, such as cleft lip palate, the congenital dislocation of the hip, congenital heart defects, and so on. Sometimes these disorders involve multiple genes, as well as environmental factors, as you can read here, for example, with the cleft lip and palate, that these are associated with dysregulation of gene expression of multiple genes, as well as environmental factors such as vitamin deficiency. Now, some of these diseases are acquired, so there is really a strong environmental contribution, like cardiovascular diseases, for example. Of course, with good diet, a person can avoid having a cardiovascular disease, but there is still genetic contribution. Okay. Now, there's some neurological diseases that it seems that these diseases are caused by multiple genes, but it's not clear yet if there is an environmental effect or not. Now, some of these diseases, by the way, or some of these genes are associated with increasing the susceptibility of a person to develop a certain disorder. So having such mutation, or let's say polymorphism, does not mean that a person would develop a disease. Rather, it just means that this person is susceptible, مستعد, to develop this disease if precautions are not considered and are not taken. Now, for example, we can have identical twins. They have the same genome, but it's not always the case that if one is affected with a certain disease, that the other one will be as well. So this means that there are non-genetic factors that are associated with certain disorders or phenotypes. Here's an example with BRCA1. So BRCA1, the name BRCA, comes from breast cancer. Now, BRCA1 was the first gene to be associated with breast cancer. And in fact, about 10% of women with breast cancer, their cancer is hereditary, meaning that they inherited a defective gene. And about 20% of them have defects or mutations in the BRCA1 gene. So now, having such mutation / polymorphism in the BRCA1 gene does not mean that a female would develop breast cancer. Rather, you can see here, for example, that you can have a female developing ovarian cancer rather than breast cancer, since ovarian cancer is also associated with uh, BRCA1 mutations as well. You can have transmission from a carrier father, for example, okay, into females. So some traits are known as quantitative traits because they are affected by multiple genes. So these are polygenic traits. So if you have, for example, a certain condition that is associated with a single gene, it would look something like this, where you have homozygous and heterozygous conditions. Now, if there is a phenotype that is affected by two genes, it would look something like this. That's the distribution of genotype in an individual or among individuals. Okay, so you would have it looking like this. Now, if there are more genes, 3 genes, then it would look something like this. Now, if there is a trait that is influenced by multiple genes, then it would look something like this, which is a normal distribution. So this is the distribution in a population where you have very few individuals having all genes homozygous at either end of the normal distribution curve. Now these traits are also influenced by the environment. Okay. Now, and here's a list of characteristics or phenotypes or disorders that are associated with that have a continuous normal distribution. And these include blood pressure, for example, which is influenced by the diet that we take, height, intelligence, body mass index, and so on. This is an illustration for genes that affect skin color. So here's an assumption that let's say that we have genes having or skin color being affected by 6 genes. Well, you can have all of these distributions of genotypes. So it looks more like a normal distribution again, and you can see from the bars how it looks like. Now, there are some guidelines that are associated with inheritance of multifactorial traits, and that is they tend to run in families. Okay, but the pattern of inheritance is not predictable. So again, having a mutation slash polymorphism does not mean that a person would show the expected phenotype. Now, the thing is that the chance of recurrence is also less than the risk for single gene disorders, and that makes sense, right? The closer the degree of relationship, the higher the risk is for a multifactorial inheritance of a disease. Okay, so this is the reason behind that is that the closer the relationship is, the more genes that are shared among individuals. So if you look at parents, siblings, children, they share half of their genomes, right? If we're talking about second degree relatives like uncles and aunts and nephews and nieces, they share 1/4 of their genotype. If we're talking about third degree relatives like first cousins, great grandparents, and so on, and great grandchildren, they share 1/8 of their genomes. So if a parent is affected, there is a higher chance that a sibling would also be affected than a niece or an aunt or whatever. Okay. Now the thing is also is that the degree of risk also increases with the degree of severity of the disorder. So if we're talking about a congenital disorder like whatever, blood pressure, for example, okay, if a father or a mother is affected by blood pressure and this blood pressure is really severe, then there is a good chance that their siblings or children would also be affected than if a person is having a mild blood pressure disorder. Something important as well is that it can be sex dependent, and we'll talk about that in a few minutes. So here's the thing. So the risk is greatest in close relatives. This is looking at certain congenital malformations like the cleft lip. Now, incidence in the general population is 1 in 1,000. If you look at monozygotic twins, the risk is this times 400. So the risk is about 40% instead of 0.1%. Now, if you look at first degree relatives, it's about 4%. Second degree relatives, it becomes closer to 1%, 0.7%. And if you look at third degree relatives, now it decreases to 0.3. It is still higher than the risk in general population, but it decreases as well. Or let's say it increases with increasing relationship among individuals affected versus related. Now, this can be explained by what is known as the liability threshold model. What this model does is that it explains the degree of susceptibility of a person to develop a certain polygenic disease or a multifactorial disease, okay, by looking at the liabilities or by looking at the factors that cause this condition to appear. So what liability indicates is all the factors that are associated with developing a disease. These factors can be genetic, they can be environmental, or they can even be pathogenic factors. So the thing is, having a certain number or combination of these factors would cause individuals to develop that condition. Okay? Now, the distribution of these factors among a population it follows a normal distribution curve. It looks like that. So most of the population do not have this condition because they do not have enough liability or factors to develop conditions. Only these people do. Okay. So here is simplification of what it talks about. So let's say that there's a certain condition that is caused by a combination of genetic factors, environmental factors, and pathogenic factors. Now, in order to develop a condition, a disease, you need to collect a combination of 5 of these factors. So you look at these different individuals in here. This person here has 2 genetic factors and 2 environmental factors and 1 pathogenic factor. The combination of all 5 of them would cause this person to develop a disease. Look at this individual. This individual has a certain different combination than this man. This boy has 3 genetic factors, 1 pathogenic factor, and 1 environmental factor, collecting 5, a total of 5, okay? And that's why this person would develop a condition. Look at this lady in here. She's truly sick. She has collected actually 7 factors, so her condition is very severe. She has 2 environmental factors, all 5 genetic factors, no pathogenic factor, but it doesn't matter. As long as she has a combination of certain liability or collection of a set of 5 factors, that is enough for this lady to develop the condition. So that's basically what it says. Now, the thing is, we can explain it, we can apply it to families. Now, notice that what happens in families is that the risk increases, right? Because they share more factors than the general population in terms of genetics, even in terms of exposure to certain environmental conditions or being affected by certain pathogens. So what happens here is that the liability, the number of factors does not change. It's still 5 factors, let's say, of whatever, environment, genetics, pathogen, whatever. But what happens is that the bell curve in here shifts a little to the right. That would increase the proportion of individuals being affected with the disease. Now, this does not change. The number of factors does not change. But what changes is that there is a shift of the curve to the right. Because because these people are more susceptible to collect these factors. Okay? So here's simplification again. There's certain conditions, the same condition. You need 5 factors: genetics, environmental factors, and pathogenic factors. And there are these 3 brothers. Now they notice that they share the same genetics in here. Okay, except that this person is affected by certain environmental factors. Okay, and that increases the susceptibility of this person to develop the disease. Well, you look at this person right here. He has collected 5 of these factors. Only 1 environmental factor, but that's fine. Now, this person has collected 7 of these factors. Okay, so this person would have a more severe condition than this person right here, and this one here is affected, but he has this person would be the least affected. But notice that all of them are affected because they share more of the genetic factors than in the previous slide, and than in individuals in the previous illustration. So there are some points that we have to consider when it comes to the threshold model, and that is risks are different from Mendelian inheritance risk. So it's not clear cut. It's not like if a person has this genotype, this person would be affected. Okay, because we have the involvement of other environmental factors that can change, by the way. So if a person is leading a bad lifestyle, this person can switch to a good lifestyle, a healthy lifestyle, and this person can reduce his or her risk of developing that condition. The second point is that risks vary among different families because they have different genetic composition. So again, it is a probability. Okay, it's not clear cut. The third point is that the risk increases with the number of affected relatives. So if you have a family having a multifactorial disorder, and there are many individuals having this condition, this means that the risk is higher than a family having very few members of that family having that condition. Probably because the first family would have more genetic components, more genetic factors, maybe even more environmental factors and more pathogenic factors that they are exposed to. And that makes sense. Now, the fourth point is that the risk increases among close relatives. It makes sense, right? Because they share the same genotype. Something else is that the differential risk to relatives decreases as the frequency of the disease increases in the general population. Okay, so the thing is, if the disease is more widespread in a population, the risk of a family member to develop a condition decreases. Why? Because there is a lower threshold that results in a smaller difference between a family of affected individuals and the population. The thing also is that the risk increases with the severity of the malformation of the disease. So if the disease is really severe, it means that the risk increases for other members of the family to develop that condition. And again, it makes sense because the severity may be related to to collecting more mutations in more genes than in people with mild disorders. Okay, so for example, there is 6% chance of for a family member to develop a bilateral cleft lip and palate than if you have uh, than if there's a unilateral cleft lip. Again, there is if someone in a family has a bilateral cleft lip, so it's more severe. Okay, there is a 6% chance that another member of the family would develop the same condition, a bilateral cleft lip. But if someone has a unilateral cleft lip, which is milder than bilateral, the risk decreases to 2%. So there is another point that we have to consider. In addition to genetic factors, environmental factors, and pathogenic factors, and that is gender. Because sometimes some disorders are associated with a certain gender. So being of a high risk gender, whether male or female, would increase the chance of inheriting this multifactorial disorder. So let's say that this father right here has the following combinations. Okay, 2 genetic factors, 1 pathogenic factor, and 1 environmental factor. In addition to being a male, this person is sick. This person is having that disorder. Now, this person right here, so the question is, what is the chance that a female member of the family would be affected and a male member of the family also being affected? Well, there's a higher chance that a male member would be affected than a female member because this condition is associated with being male. So this person would have the maleness factor associated with the disease, or helping this person or facilitating this person in developing this disorder. Notice that this female right here, the sister right here, let's say, she has the same genetic component as both of them, but she doesn't have the gender factor. And this can be illustrated right here in this condition, the pyloric stenosis, where you have narrowing of the canal that links the end of the stomach with the duodenum. Now, the thing is, if the father is affected, there is a higher chance that his sons would be affected than his daughters. Okay? Now, well, what if the female is affected? Okay. What is the chance that a male would be affected and a female would be affected? Again, the disease is associated with being a male. So the thing is, there is a higher chance that both males and females would be affected. Okay, because the female here is affected, so she has more of these factors other than being male. She has more of these genetic factors, environmental factors, pathogenic factors that led to the development of the disease. So this person here has a high chance of developing a disease because in addition to having the same genetic contribution or environmental contribution and pathogenic contribution, being male would make it even worse. So you would have more males in the family being affected, and you will also have females at risk, but males would have higher chance of being affected. Okay, so looking here at the same table, if the mother is affected, boys and girls would be affected. But notice that the probability that boys would be affected is much higher than if the father is affected. Okay, and the risk of females being affected is also higher than if the father is affected. But still, remember that being male is associated with developing the disease. Okay. Also, look at the population. The risk is higher also for males than females as well. So let's apply what we have just talked about to the threshold model. That is the sex-dependent inheritance of a multifactorial condition. Now, notice here, we have the same table. We have individuals having pyloric stenosis, and we said that it is male dependent. So males are more affected than females. If the father is affected, okay, so if the father is affected, there is a higher chance that sons, that males in the family would be affected than females. Okay, so if the mother is affected, remember now, it's still a male dependent condition. So if the mother is affected, males are still more affected than females, but notice the proportion of males that are affected is much higher, as well as the proportion of females that are affected. That's because, again, mothers, in order for females to develop a condition, they have to have many factors, many more factors than males. And if they have these factors, then there's a higher chance that these factors would be inherited to siblings, to offspring, I'm sorry, to their offspring. And males, again, being males, would be more affected than females. So let's apply this to the threshold model shown in here, but I'm going to go to the next slide, and you can read what's written on this slide. So here we have, again, the liability on the x-axis and normal distribution of the liabilities, or the factors that are necessary for the development of this multifactorial condition. Now, you have a certain number of factors, right? Still the same. And notice that you have more males in here affected than females. Females are shown in here. So the proportion of males that are affected is much higher than females, being a male dominated or a male dependent condition. Now, that's in the general population. Now, let's look at the curve if a male is affected in a family, and let's look at the risk of other individuals in that family being affected. The curve, again, would shift to the right. The same threshold applies, so you can go back to the previous slide and look at the drawings. Now, it's the same number of factors, but what happens is that you have more males affected. The proportion of males is much higher, as well as the proportion of females. Yet still, males are more affected than females. Okay, so what if, let's look at the curve, if a female is affected. Again, there's a shift to the right. Now the proportion of individuals being affected is much higher, and males are still more affected than females. So let's look at another concept, and that is heritability. That is, we want to know what is the extent of the genetic contribution to a multifactorial condition, trait, or disease even. So there are many ways by which we can apply, or we can study the genetic contribution of a certain condition. Probably one of the best, the oldest and best, is twin studies. The idea is that now monozygotic twins have identical genomes, right? versus dizygotic twins, non-identical twins. So the thing is, if a condition is truly 100% genetic, then monozygotic twins will be 100% concordant than dizygotic twins versus dizygotic twins who will have a lower concordance rate. So what does concordance means? That is a match. So there, if one is affected, the other one must be affected. If the condition is 100% dependent on genetics. Okay, so there would be higher concordance, higher correlation. If a trait is 100% environmental, then monozygotic twins and dizygotic twins will have the same concordance rate. Okay, because there's no genetic contribution. There should be no difference between monozygotic twins and dizygotic twins in terms of being carrying this phenotype. So let's look at this. This is a really nice illustration. Look at certain conditions here. Rickets, and that's the concordance rate. That is, if one is affected, what is the chance of another one being affected for both monozygotic twins and dizygotic twins? So if you look at rickets, for example, which is a genetic disease with some environmental contribution. Now, if you look at it, for example, if one monozygotic twin is affected, then there's an 80% chance that the other monozygotic twins would be affected as well. Okay, so how about dizygotic twins? Well, if one is affected, the chance that the other one is affected is about 20%. So it's much less than monozygotic twins. So you would say, aha, there is a genetic contribution now. Well, let's look at something else. Let's look at measles. Okay, now measles. If one monozygotic twin is affected, then there is about an 85% chance that the other one would be affected. Great. Well, how about dizygotic twins? Again, if there is an 80% chance that if one is affected, the other one would be affected. So there is really no difference between dizygotic twins and monozygotic twins because it's contagious. Okay, it has nothing to do with genetics. Let's look at alcohol drinking now. There is 100% concordance. Well, how about in monozygotic twins? How about dizygotic twins? There is an 80% concordance. What does that mean? It means that there is probably some genetic contribution, but environmental conditions are more important, more significant. Okay, let's look at something else. Stomach cancer. Okay, if one is affected, then there's a 20% chance that the other is affected in monozygotic twins. How about dizygotic twins? It's about 5%. So this indicates that there is a genetic contribution. Okay. You can look at eye color as well. Okay. If one has a certain eye color, then the other one would have, there's a 95% chance that the other one would also have the same eye color. Okay. Versus dizygotic twins, it's much less, which means that there is again a genetic contribution. So how can we calculate concordance and heritability? So the concordance rate is calculated for both separately. So the idea is that we take if both are affected over the total number of twins studied. That is one affected plus both are affected times 100. So that shows the concordance rate for both monozygotic and dizygotic twins. Now, if there is if a trait is 100% determined by genetics, then it should be closer to 100% in monozygotic twins and 0.5 in dizygotic twins of the same sex again. So to measure the genetic contribution, or to measure the genetic contribution, you take the ratio, concordance rate for monozygotic twins over concordance rate for dizygotic twins. The higher the number, the more genetic a trait is. Okay. Now this is for calculating concordance ratio. How about heritability? There are several formulas. One of them is this in here. Heritability, that is the genetic contribution of a trait, is 2 times the concordance rate for monozygotic twins minus the concordance rate for dizygotic twins. Okay. Again, so you can do the same calculations on these to determine the heritability of these conditions. Okay, so you can convert this into numbers. So let's go over some characteristics of multifactorial inheritance. One, although the disorder is familiar, there is no distinctive pattern of inheritance. So it's not clear-cut again. It's not like you say, this person has these genetic polymorphisms or whatever, then this person is destined to be affected by that condition. The risk to first-degree relatives determined by family studies is approximately the square root of the population risk. So the idea is that if there is a certain condition and it exists in 1 to 1,000 in a population, then first-degree relatives would have a 3% chance of developing that condition if it exists in that family, or if a first-degree relative is affected. So there's 3 in 100 chance rather than 1 in 1,000 chance of that person to be affected with that condition. The risk is sharply lower for second-degree relatives than for first-degree relatives, and it makes sense because they share less genetics or less of the genotype, genetic composition than first-degree relatives. The recurrence risk is higher when more than one family member is affected, because that makes sense again, because they share more genes associated with developing that condition. And of course, the more severe the malformation, the greater the recurrence risk, because the more mutations or polymorphisms exist in an individual, and these mutations or polymorphisms associated with that condition, the more severe the case would be. If a multifactorial trait is more frequent in one sex than in the other, then the risk is higher for relatives of patients of the less susceptible sex. So you can go back to the sex-dependent slides that we just talked about, or conditions, sex-dependent conditions in the slides that we just talked about. An increased recurrence risk when the parents are consanguineous suggests that multiple factors with additive effects may be involved. So that's just again genetic contribution, more genetic contribution.